In this video we will discuss whether the enzyme activity, enzyme means glycosyltransferase or this sialyltransferase, are responsible for reducing inflammation, or it's the glycan structures that occurs because of these enzymes are responsible for reduced inflammation. Before this paper has been published, there has been some reports that it's the glycan structures that is responsible for reduced inflammations. So in this video we will discuss the result where they have tested whether it's the enzyme that is responsible for reduce inflammation or it's the glycan structures that is responsible for reduce inflammation. In order to check this hypothesis, what they did is they mutated this sialyltransferase at two different positions, that is C350A and C350 is replaced with A and C361 replaced with A, and this mutated enzymes they called it as ST6 Kaka, or the combined co-culture of this B4F6 and mutated ST6FC is called as B4ST6FC Kaka. And when they use these enzymes, they check the inflammations and compare it with the control, that is PBS, they are able to see that when they use this B4ST6FC Kaka, the inflammations does not goes down, and it is similarly, and it is in line with the results that are obtained for the control, that is PBS. Whereas for the other case such as IVIG and B4ST6FC, they see the reduced inflammations. And one more thing that they have checked, that this mutated ST6-Gal1 Kaka has the reduced activity, but it does not affect the sialyltransferase function of this enzyme, and this results has been shown in the supplemental figures. You can refer to that figure to check whether it will affect this mutated IgG will affect the sialyltransferase or sialic acid transferase activity functions or not, but it will affect the enzyme activities, and both the things have been tested in the supplemental figures. You can check that figure to understand how they have made this point. So, and the quantifications of this panel B is shown in the panel C, where we can see that when the Kaka, this B4ST6FC Kaka is used, the inflammations are in line with the inflammations, that is the results that are obtained for the control case, that is with PBS. So from this they come up to a point that the glycan structures, even though it is present, they are not responsible. It doesn't seem to be responsible for this reduced inflammations. So next, what I did is they check the functions, they check the effect of inflammations in the absence of glycan structures and the active enzymes. So in this case, what they did is they use the transferase, that is endotransferase, that can cleaves off that can cleaves off the sialic acids. But in this case, they do not use the mutated IgG, but they use the wild type IgG combined with this, and they use the co-culture of B4 engineered IgG and ST6 engineered IgG together. And what they find out is that even though the sialic acids are absent because of the presence of these enzymes, are cleaved off, not absent, are cleaved off because of the presence of this enzyme, they see the reduction in the inflammations that can be seen here, that for the PBS the clinical scores are high, whereas for the case where B4ST6FC and endo-TH is used, where there is no sialic acids or sialic acids are cleaved off by these enzymes, they see the reduced inflammations. And these results are also in line with the control case where B4ST6FC both are used and without any enzymes and with just the endo buffer. And also these results are in line with the another control case where this B4ST6 FC is used. So this indicates that, and the quantifications of this panel E is in the panel F, where we can see that where we can see that reduction in the clinical score for all the cases, that is with the when they use IVIG, or IVIG results are also shown in the panel E. So when the results for the IVIG and B4 S6FC or B4 ST6FC with the endo buffer, or B4 ST6FC with endo TX are non-significant, but they are significant compared to the control case, that is PBS. So combining both the results, the researcher says that it's the enzymes activity that is responsible for reduced inflammations rather than the glycan structures. So I hope you understood how they come up to this point from this results that are presented in this figure. If you need to understand more about this experiment in detail and need to refer to the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.